Good morning, everyone. Today we will start the main metabolic pathways related with fatty acid metabolism. So, as you know, that if we see the importance of fatty acid, it is useful. Oxidation of fatty acid gives us the energy, just like glycolysis and TCA cycle. Also, fatty acid is a reservoir, fat reservoir in the form of triacylglycerol. It helpful during the adverse conditions such as the starvation or if there is any error with the carbohydrate metabolism. Fatty acids are also helpful in synthesis of phospholipids, sphingolipids, cholesterol esters, and prostaglandins, etc. etc. Normally, free fatty acid level in the blood is between 100 to 200 milliequivalents per liter. Higher level can be seen during the starvation or uncontrolled diabetes mellitus because in both the condition the carbohydrate amount will be low. So there will be excess of fat breakdown for the energy purpose. Fine. So most important pathway in fatty acid metabolism, which can be asked in the form of short note, is beta oxidation of fatty acid. So you can compare it with the glycolysis of carbohydrate metabolism. There are various other form of oxidation of beta acids. They are alpha, omega, perioxisome, and unsaturated fatty acid oxidation. Out of this one can be asked in the form of short note or even in the form of 10 marks question. Other oxidation, you don't have to remember the pathway, you just have to remember the disorders, which can be asked in 2 marks. Also in the Viva verse. Okay, fine. So beta oxidation of fatty acid. First, if we want to define what is beta oxidation. So it is the process by which there will be generation of two carbon compound acetyl CoA by oxidation of beta carbon of fatty acid. So here our substrate will be the fatty acid, the product will be the acetyl CoA, which is a two carbon fatty acid. And this acetyl CoA will be going towards TCA cycle and will give us the ATP. Okay. Similarly, in case of carbohydrate, glucose underwent the process of glycolysis. There was generation of pyruvate. Pyruvate was getting converted to acetyl CoA by PDH complex. So here there is no intermediate like glycolysis. Directly fatty acid will be oxidized into acetyl CoA and then there will be generation of ATP. Okay. So main thing here is the oxidation occurs at the beta carbon of fatty acid. If we see the major organs, so it takes place in most of the organs, metabolic organs, liver, kidney, heart, also adipose tissue, etc., etc. Subcellular site includes inner mitochondrial membrane and matrix. Also, initial part takes place in the cytosol. So this is also you can remember that this pathway also operates at the both the site, cytosol as well as in the mitochondria. So first we have to understand or recall what is beta carbon. So beta carbon, if you can remember the chemistry of fatty acid, okay, or the classification of fatty acid where we have given certain numbers alpha, beta, gamma to the carbons. So the carbon which is adjacent to the carboxylic group is known as the alpha carbon, and the carbon which is next to alpha carbon is known as the beta carbon. Okay, so we can say that the beta carbon will be two carbon away from the functional group, that is the carboxylic group. Okay, this is another way of numbering. If we give carbon number 1 to the carboxylic group, then adjacent carbon will be carbon 2. The next will be the carbon 3, which is also known as the beta carbon. So here in this process, there will be oxidation of this particular carbon. So majority of the fatty acids, almost we can say 70 to 80% of fatty acids gets oxidized by beta oxidation. Okay, so for that you should know what is a beta carbon. So the final product of beta oxidation of even chain fatty acid, even chain, that means for the fatty acid which contains carbon 2, 4, 6, 8, etc., 16, for them the final production will be, or final product will be acetyl CoA. In case of odd chain fatty acid, example is propionic acid. In that, the end product is propionyl CoA, which is a 3 carbon compound. Acetyl CoA is a 2 carbon compound. Okay, so if you can see this classification, acetic acid will have 2 carbon, propionic acid 3 carbon. So whatever other fatty acids which are mentioned here, okay, if they are having even carbon, then the product will be acetic acid or acetyl CoA. Similarly, in case of other carbons such as propionic acid, other fatty acids such as propionic acid which are having odd number of carbons, for them the end product will be propionyl CoA. Okay. So small and medium chain fatty acid can directly enter into the mitochondrial membrane, which are mostly present in the milk. But for long chain fatty acids, okay, which is usually present in our diet, they require a special transport system to enter into the mitochondria. Okay, so for the movement of cytosol to mitochondria, where actual oxidation takes place, long chain fatty acid requires this particular transport system, carnitine transport system. In case of small and medium chain fatty acids, they can directly enter into mitochondria. Okay, please remember this point. It will be helpful in understanding the certain disorders. Okay, so first step, the fatty acids which are there in cytosol, they has to be activated. So this is a fatty acid. Okay, you don't have to draw any structure, you just have to understand the structure for the better understanding of metabolism. Okay, so first fatty acid has to be activated into fatty acyl CoA. So there will be addition of coenzyme group, which is a derivative of pantothenic acid. Okay. So pantothenic acid derivative coenzyme A, along with that there will be addition of sulfur group. So coenzyme A SH has to be transferred to fatty acid. So the product will be fatty acyl CoA. Okay, fatty acyl CoA, or sometimes it is also written as fatty acyl CoA SH. Okay, for this process, or for to add this particular group, there is a requirement of energy. So an ATP will be utilized here. This ATP will be hydrolyzed into AMP and pyrophosphate. So always remember, in any metabolism, if you see this kind of reaction where ATP, instead of getting converted to ADP, if it is directly getting converted to AMP and pyrophosphate, the amount of energy which is required is equivalent to 2 ATP. So here we have to hydrolyze two bond, okay, two phosphoesterase bond. Then direct conversion of ATP to AMP is possible. Okay, so though it is written as single ATP, in real scenario, the amount of energy which is utilized to catalyze this reaction is equivalent to 2 ATP. So here 2 ATP are utilized in this particular first step where fatty acids are getting activated. Okay. Now remember, this is for only long chain fatty acids. Small and medium chain fatty acids, they can directly enter into mitochondria without the need of any other thing. Okay. So here, this is the first step where activation of fatty acid takes place in the form of formation of fatty acyl CoA. It is similar to your glycolysis first step, where glucose or any carbohydrate has to be converted to its phosphorylated form, glucose 6 phosphate or fructose 6 phosphate, for the further metabolism. So similarly here, fatty acid is converted to fatty acyl CoA. The enzyme which is required here is acyl CoA synthetase, and another name of this enzyme is thiokinase. Okay, another name of this enzyme is thiokinase. Fine. Second thing is that now the fatty acid has been activated. To move from cytosol to mitochondria, it requires a transport system which is known as the carnitine transport system. So what is a carnitine? So carnitine is actually a product of lysine and methionine. Okay, lysine and methionine combines and forms the carnitine. Chemically, it is beta hydroxy gamma trimethyl ammonium butyrate. So no need to remember this kind of complicated name. Just remember it's a derivative of lysine and methionine. Also, it is present in certain meat products. It is usually abundant or getting stored into the skeletal muscle. Deficiency of this will lead to impairment of fatty acid oxidation, because fatty acid will not be able to transfer into mitochondria, so the oxidation gets stopped. Okay. Also remember, since it is synthesized from lysine and methionine, so it also helpful in collagen formation. So carnitine tablets are available, and they are usually prescribed if there is, in case of soft tissue injury, to improve the healing process. Okay, fine. So how does this carnitine transfers fatty acid from cytosol to mitochondria? So first, our fatty acid has been activated into cytosol. So first, from outer mitochondrial membrane to intermembrane space. Intermembrane space. First, we have one transporter which is known as the CPT1. CPT1, that is carnitine palmitoyl transferase 1. It directly transfer activated fatty acid or fatty acyl CoA from outer mitochondrial membrane to intermembrane space. Okay. Now the main transport system includes CPT2, that is carnitine palmitoyl transferase 2. Okay. Sometimes some of the books also gives this as carnitine acyl transferase 1 and carnitine acyl transferase 2. So both are same names, carnitine acyltransferase or palmitoyltransferase. Basically, they will transfer the fatty acid from outer mitochondrial membrane to inner mitochondrial membrane and towards the matrix side. So this CP2, what does it do? The fatty acyl CoA, which we have got from outer mitochondrial membrane, the carnitine, which is a transporter, this fatty acyl CoA from its structure, the fatty acyl or acyl group will be transferred towards the carnitine. Okay, and coenzyme A will be liberated. Okay, so acyl group will be transferred from fatty acyl CoA to carnitine. This carnitine will now go towards the mitochondrial membrane, inner side of mitochondrial membrane towards the matrix side. Okay, so this acyl carnitine, after getting into the matrix with the help of same translocase or transferase, now there will be transfer of this fatty acyl group to coenzyme A, which is already available in mitochondrial matrix by certain other pathways. Okay, so acyl group from carnitine will go towards the fatty acid, and carnitine now once again will be available for transfer of another fatty acid. Okay, so fatty acyl CoA from its structure, fatty acyl group will be transferred to carnitine. So carnitine will be known as acyl carnitine. This acyl carnitine will transfer towards the matrix. In matrix, the acyl group will be transferred from carnitine to fatty acid. So we have our fatty acyl CoA, and remaining carnitine part once again goes towards the intermembrane space. So net effect will be of this carnitine shuttle is that long chain fatty acid CoA is transferred from outside to inside mitochondria. Okay. Remember, carnitine shuttle or carnitine itself is a 2 marks short note. So in that you don't have to write anything. You just have to draw this particular shuttle. Now, our fatty acid, which is the active form, that is fatty acyl CoA, it has entered into the matrix of mitochondria. So now the actual process of oxidation is getting started. Okay, so the first enzyme in this process we already mentioned, that is thiokinase. Second enzyme which is necessary here is the dehydrogenase. Okay, dehydrogenase. In this, as a cofactor, FAD is required. So if we see the first process here, fatty acyl CoA is getting converted to enoyl CoA. Okay, another name of this is alpha beta unsaturated fatty acyl CoA. Okay, alpha beta unsaturated fatty acyl CoA, or you can remember as enoyl CoA. So here what happens? There will be transfer of hydrogen from this to carbon to FAD. So FAD will be converted to FADH2, and oxidation of fatty acid is getting started. So fatty acyl CoA will be oxidized into enoyl CoA. In this process, the FAD will be reduced to FADH2. After that, there will be addition of H2O. So enzyme name will include hydratase. Okay. In first one there was dehydrogenase. Now there is hydratase. Okay. So after addition of this hydratase or hydrogen, okay, if you can see here, there is a addition of this OH group at the beta carbon. So the product will be hydroxyacyl CoA. Okay, so the product is hydroxyacyl CoA. Okay, just after addition of OH, you can remember hydroxy acyl CoA. One more thing I want to tell you is that here it is written as it's a family of chain length specific enzyme. So this dehydrogenase will be different for small chain fatty acid, for medium chain fatty acid, and for the large chain fatty acid. So dehydrogenase will be specific to corresponding length of fatty acid. So if there is a defect of any particular dehydrogenase enzyme, then oxidation of only that length of fatty acid will be affected. So example, if there is a deficiency of medium chain fatty acyl CoA dehydrogenase defect, then medium chain fatty acid will be difficult to oxidized. Okay. So after this enoyl CoA and formation of hydroxyacyl CoA, once again there will be process of dehydrogenase. So the hydrogen will be removed from the beta carbon and it will be transferred to NAD. So NAD will be converted to NADH2 and our beta carbon, if you can see here, now has lost this second hydrogen also. So the product is ketoacyl CoA. Okay, try to remember these three names. They are a bit difficult to remember, but if you see the process here, first there is a dehydrogenase, that is removal of hydrogen, then there is addition of water, once again there is removal of hydrogen. Okay, so enzyme names, even you can remember only dehydrogenase, hydration, and dehydrogenase. Okay, but this product name you have to mug up somehow. Okay, so first fatty cell CoA converts to enoyl CoA. In that process FAD will be reduced to FADH2, then there will be addition of water, so there is formation of hydroxyacyl CoA. Once again there is action of dehydrogenase, then there will be formation of ketoacyl CoA. After this, the final steps takes place. So if you see here in final step, enzyme thiolase on addition of coenzyme A, on addition of coenzyme A, it will release the final two carbon of this fatty acid in the form of acetyl CoA. Final two carbon of this fatty acid in the form of acetyl CoA. Remaining portion of fatty acid, which is a fatty acyl CoA, will go process of further oxidation. So this cycle once again will be repeated. So how to understand this process? So suppose if this is a 16 carbon fatty acid, for example, palmitic acid. Okay. So in palmitic acid, during the first cycle of beta oxidation, our product acetyl CoA will be released. It will go towards the TCA cycle. Remaining 14 carbon compound or fatty acid once again will go towards the oxidation process. So from that 14, another 2 carbon will be released and remaining 12 will go for the beta oxidation. Okay, so this process will continue until we have only 2 carbon compound, that is acetyl CoA, is left. Okay, so try to remember the name of these 5 enzymes. First is thiokinase, then dehydrogenase, hydration, dehydrogenase, and thiolase. Okay, so how to calculate the energetics? So if we take an example of palmitic acid, which is a 16 carbon compound. Okay, so during the first process of beta oxidation, okay, this 2 carbon will be released in the form of acetyl CoA. Remaining 14 will go under the process of beta oxidation. From this 14, these two will be repleted remaining will be the 12 carbon. Another beta oxidation cycle, these two will be released in the form of acetyl CoA, remaining will be the 10. Similarly, we have 8, 6, 4. Okay, now this is the final oxidation step. Here what happens once the fatty acid chain is reduced to 4 carbon, then from 4 carbon the process of beta oxidation will release one molecule of acetyl CoA, which is 2 carbon. Okay, but the final remaining part will be a 2 carbon compound, fatty acid derivative, 2 carbon compound, which itself is acetyl CoA. Okay, so this will not have beta carbon, and this acetyl CoA can directly go for the TCA cycle. Okay, so the final oxidation step will give you directly, when you convert C4 into C2, you will have 2 acetyl CoA. So the complete oxidation of this palmitic acid, what it will produce, it will have total 7 cycles. Okay, we'll have 7 cycles. If you can calculate, this is the 1, 2, 3, 4, 5, 6, and 7 cycle. But in the 7th cycle, we will have total 2 acetyl CoA. Okay, so total we have got how many acetyl CoA? We have 8 molecules of acetyl CoA. Okay, 8 molecule of acetyl CoA if the chain is of 16 carbon. Okay, fine. But the beta oxidation is taking place only 7 times. So if you can see the pathway of beta oxidation, here we get one FADH2, one NADH2. Okay, here this process, these two are the step which gives us the reducing equivalent. Okay, so for total 7 cycle we get 7 FADH2, 7 NADH2, and 8 acetyl CoA, because last step gives us the 2 acetyl CoA. Okay, fine. So if you want to calculate the energetics, so according to old calculation, 1 FADH2 can gives us the 2 ATP, so the 7 will give us 14. 1 NADH2 gives us 3 ATP, so it is 21, and 8 molecule of acetyl CoA goes towards the TCA cycle and gives us the 12 ATP. So 12 into 8, 96 ATP. Okay. But according to new calculation, you have to remember that 1 FADH2 gives us 1.5 ATP. So 7 FADH2 will give us 10.5 ATP. 7 NADH2 will give us 17.5 ATP. Okay, you have to remember 7 into 1.5, here 7 into 2.5. Okay, and for the Krebs cycle we will have 80 ATPs depending on new calculation. So total how many ATPs we get? 108. Okay, we get 108 ATP, but remember we required 2 ATP for fatty acid activation. Okay, so the net production or net ATP production will be 106 ATP in case of palmitic acid. Okay, remember for 16 carbon fatty acid oxidation we get 106 ATPs. Okay, similarly it can be asked, question can be asked in case of 14 carbon fatty acid or 18 carbon fatty acid. So you have to do your calculation accordingly. So based on the carbon number, either one cycle of beta oxidation can be increased or reduced. Depending on that, final calculation will differ. But most commonly, it is asked for the 16 carbon, that is palmitic acid, where you get net 106 ATP. Okay, so this much amount of energy difference is produced in compared to carbohydrate metabolism, where in case of aerobic glycolysis we were getting net 8 ATPs. Here it is 106 ATP. Now coming to the beta oxidation of odd chain fatty acid. So odd chain fatty acid produces propionyl CoA and acetyl CoA as its final structure. How? So suppose if we take an fatty acid example of fatty acid which has 17 carbon, it will go 7 cycle of beta oxidation. So it will produces 7 acetyl CoA. Remaining 3 carbon compound will be directly released in the form of propionyl CoA. Remember, example of odd chain fatty acid is propionic acid. Okay, and 2 carbon fatty acid is acetic acid. Okay, so 7 into 2, that is 14, and remaining 3 carbon will give us the propionyl CoA. Okay, so for odd chain fatty acid, the product is propionyl CoA, and for even chain, it is acetyl CoA. Process is similar, just like beta oxidation. Uh, Only change final three carbon compound will be directly released. The acetyl CoA, as you know, will go towards the TCA cycle. But what about propionyl CoA? Okay, propionyl CoA, first it will be converted to methyl malonyl CoA, and methyl malonyl CoA will be going towards succinyl CoA. Methyl malonyl CoA will produce succinyl CoA. Okay, this reaction is catalyzed by vitamin B12, and you have already learned this reaction in function of vitamin B12. Okay, vitamin, this is one of the only two reactions which is getting performed with the help of vitamin B12 in our body. Okay, so the propionyl CoA forms an intermediate of TCA cycle, or it enters into TCA cycle via succinyl CoA. Regulation part. So there are two regulatory steps here. One is the CPT1 transporter. Okay, that is carnitine palmitoyl transferase 1 transporter. So malonyl CoA, which is an intermediate of fatty acid synthesis, okay, which we will learn when we discuss about fatty acid synthesis, it inhibits this particular transporter. So here what happens when there is a well-fed condition, when glucose is more, then there will be more production of pyruvate, more production of acetyl CoA, and more production of fatty acid. Okay, acetyl CoA will produce fatty acid. So what does it mean is that we have sufficient amount of energy from glucose, so there is no need of fatty acid to get oxidized, because it will be just a waste of energy. So body, what it will do, if we have sufficient energy from other sources, say glucose, then more fatty acid will be synthesized from it, and one of the product or intermediate of this fatty acid synthesis pathway is malonyl CoA. It will, its concentration will be more, and it will inhibit the CPT, so no beta oxidation. In contrast, in starvation, where all this will be very low, in case of starvation, so at that time there will be low production of malonyl CoA, so there will be no inhibition of CPT, and there will be more oxidation of fatty acid for ATP production. Okay. Another thing, if there is a sufficient amount of energy or ATP is sufficient, then ATP and ADP ratio will be more, or NADH to NAD ratio will be more. They will inhibit the beta hydroxyacyl CoA dehydrogenase enzyme. Okay, so these two are the regulatory step of this particular pathway. Coming to the defect of beta oxidation. Okay, all these defects are separately 2 marks questions. So in that, the you have to remember the deficient enzyme name. Okay, so the first defect is the medium chain fatty acyl CoA dehydrogenase defect or deficiency. Another disease is Jamaican vomiting syndrome, and third one is methyl malonic aciduria. So first one, okay, medium chain fatty acyl CoA dehydrogenase deficiency. So as I told you, during the beta oxidation process, the first acyl CoA dehydrogenase will be separately for small chain fatty acids, separate one for medium chain fatty acid, and separate one for long chain fatty acid. So here the fatty acyl CoA dehydrogenase, which is specific for medium chain, will be defective, or there will be deficiency of medium chain fatty acyl CoA dehydrogenase. So in that case, what happens? Usually, medium chain fatty acids are more in milk, particularly in the breast milk or even the milk products. It will be more in this one. Milk products such as the paneer, okay, or also it is small quantity present in animal fats, etc. So there will be difficulty in digestion of this food, this type of food, milk and milk products. So defect of this enzyme can lead to a disease which is known as the sudden infant death syndrome. What will be the clinical features? Clinical features will include hypoglycemia, that will be low blood sugar, vomiting, lethargy, coma in severe state. Also, the hypoglycemia will be non-ketotic. Treatment will include high carbohydrate diet and carnitine supplements. Okay, both of these things will increase the beta oxidation process. So here, if you want to remember the symptoms, so any deficiency of any fatty acid will produce common type of symptoms such as the vomiting, lethargy, coma, hypoglycemia. So lethargy or weakness we can understand because there will be low amount of ATP. So there will be low energy. Okay, because ATP which are supposed to be produced will not be produced. Other symptoms will be kind of coma or hypoglycemia. That is because if we don't have fat for energy purpose, our other two nutrients will be utilized excessively. So glucose and protein both will start to break down for the purpose of energy. Okay, so more amount of glucose if it is utilized, it can produce hypoglycemia. More amount of protein if it is getting broken down, then there will be more production of ammonia. And in ammonia toxicity, you already know it can lead to hepatic encephalopathy, which can produce coma or vomiting. Okay, it can also hamper the electrolyte imbalance. Okay, so that can also contributes towards the vomiting. Okay, so the symptoms you can remember logically, that if fatty acid is not available, then our other source of nutrients, that are the carbohydrate and fat, will be utilized excessively, and that can lead to certain complication. Another thing is the non-ketotic hypoglycemia. So so there will be no ketone bodies. Why no ketone body formation? Because ketone body is a product of acetyl CoA. And since fatty acid oxidation is getting compromised, so acetyl CoA will not be in that much amount so that it can produce more of ketone bodies. Okay, so that's why it is non-ketotic hypoglycemia. Also, there is a role of hormones such as the insulin glucagon that we will discuss when we talk about the integration of metabolism. Second important disease is the Jamaican vomiting syndrome. Okay, the name is given because of its regional distribution towards the western countries, particularly near the sea coast, where there is a high consumption of this ackee fruit. Okay, ackee fruit. This ackee fruit contains a toxin which is known as the hypoglycine. This hypoglycine inhibits our acyl CoA dehydrogenase enzyme, and this it can produce the impairment of beta oxidation, can lead to hypoglycemia, vomiting, convulsion, and coma. Okay, so here you have to remember the word ackee fruit and hypoglycine, which can lead to Jamaican vomiting syndrome. Third one, methylmalonic aciduria, which you have already learned in vitamin B12 deficiency. So if there is a deficiency of vitamin B12 or mutase enzyme, then conversion of methylmalonyl CoA to succinyl CoA is blocked. So there will be higher production of methylmalonyl CoA, which usually gets excreted in the urine. Okay, so that is the thing. Symptoms will be the same like acidosis, CNS damage, growth impairment. Treatment will include avoid odd chain fatty acid and give vitamin B12 supplement. Why odd chain? Because odd chain will produce propionyl CoA, and from propionyl CoA we get all these compounds, methylmalonyl CoA and succinyl CoA. Then you can also have the carnitine defects. Okay, in carnitine defects, there can be defect of CPT1 or CPT2. In CPT1, defects affects the liver. It is a genetic defect. If there is a defect in the codons which are coding for CPT1, then there will be deficiency of CPT1. So mainly large chain fatty acids, its oxidation will be impaired. Also, there will be symptoms like hypoglycemia, coma, and if it is in severe amount, can lead to infant or children death. CPT2, here it is mainly related with the cardiac and skeletal muscle defect because mainly the carnitine is present in this type of skeletal muscles. So here there will be muscular weakness or can lead to cardiomyopathy, particularly after severe workload. So in treatment, we have to avoid the prolonged fast. We have to adapt to high carbohydrate diet, low to or diet which is low in long chain fatty acid, and some of the supplements can be given. Okay, so basic concept here regarding symptoms or treatment will be the same. Symptoms I have already described. In treatment, we have to depend on the supplements of fatty acids and carbohydrates. Okay, fine. In case of secondary, we will have a disease of liver, okay, such as liver cirrhosis. Because of that, there will be defect in synthesis of carnitine. Sometimes malnutrition, severe infection where excessive protein breakdown takes place. At that time, the carnitine will be low. Okay, so the long chain fatty acid can't be utilized as an energy source. Okay, so that was about the beta oxidation and their related disease. Few other varieties of oxidation we will discuss in brief. That is alpha oxidation. So in case of branch chain fatty acid, what happens here? The beta carbon is masked by methyl group. Okay, so that will prevent beta oxidation. So in this kind of fatty acids, the oxidation of alpha carbon takes place and there will be release of this carboxylic group at each of end of each cycle. Okay, so that it will be the oxidation of alpha alpha carbon of fatty acid. Also, these kind of fatty acids are present in milk, butter, and fish, commonly known as the phytanic acid. Okay, phytanic acid undergoes alpha oxidation. Okay, here remember there will be no production of ATP. Okay, so some of the dietary compounds will produce or will go under process of oxidation by alpha oxidation. Okay, we are not going in more detail. You just have to remember in alpha oxidation that branch chain fatty acid and phytanic acid, which is present in milk, butter, and fish, goes to under the process of alpha oxidation. The enzyme here, okay, will be phytanic acid hydroxylase. Okay, this is the main enzyme. Now we have a disease known as the Repsom's disease. Deficiency of this phytanic acid hydroxylase, okay, will lead to Repsom's disease. So here what happens? There will be accumulation of phytanic acid, which is we are getting from the milk products. Mostly, it is seen in early childhood or adulthood. So do remember that efficient enzyme of Repsom's disease, which is can be asked in the Viva, can be 1 mark of spot exam, or can be a shortener of 2 marks. This question was asked recently even in the NEET PG exam. Okay, the deficient enzyme in Refusum disease is. Then there were four options. The right options was phytanic acid hydroxylase. So since there will be defect in the fat soluble vitamins formation, there are chances that the clinical feature will include something like night blindness, cataract, or retinitis, all related with the eye. Similarly, neurologically can be neuropathy, neuronal deafness, or cardiomegaly also can be seen. Diagnosis can be made by higher level of phytanic acid in the blood as well as the urine. Treatment will include restriction of the products which contain high amount of phytanic acid, and last option will be the plasma pheresis. Omega oxidation, it is a very minor pathway. So all of you know that last carbon of the chain is known as the omega. Okay, oxidation if starts from this one, it is known as the omega oxidation. It will give rise to dicarboxylic acid. Okay, final type of oxidation is peroxysomal oxidation. So peroxisome, you know that it is the organelle present in the cell which is having a certain enzymes such as the catalase and peroxidase, which prevent us from the free radical injuries. Okay, so here fatty acid or amino acid oxidation produces H2O2, which is destroyed by the catalase. Some very long chain fatty acid and phytanic acid undergoes oxidation by in this organ, that is perioxysome. But what is the main difference here? After the first step of, that is fatty acyl dehydrogenase, here is that electron from FADH2 will be directly transferred to oxygen and it forms the H2O2. Normally FADH2 goes towards the ATC and produce ATP, but the difference here is that in perioxysome FADH2 will transfer its these hydrogens to oxygen directly. So there will be formation of H2O2. Okay, the defect associated with perioxysome, first ferrioxysome beta oxidation is Zellweger syndrome. So here what happens? There will can be congenital absence or decrease in the function of perioxysome because the gene, that is peroxin, which is necessary for the formation of peroxisome. Peroxisome. Okay, so that will be in low amount or there can be absence of this. So there will be no peroxisome. So very long chain fatty acids, means fatty acids which are more than 26 carbon, they will be difficult to get oxidized and they either will be accumulated or Sometimes it can be accelerated. Accumulation of this kind of fatty acid can produce neurological features. Okay, so here Zellweger syndrome, the defect of the gene will be peroxisome. Okay, remember this question was also asked in the NEET PG exam. Another disease is X-linked adrenoleukodystrophy. Here there will be defect of transport defect. So long chain fatty acid cannot enter into the peroxisome. The feature will affect adrenal gland and there will be neurological features. So here the protein which is getting mutated is the star protein, that is steroidogenic acute regulatory protein. Okay, so this also can be a potential NEET PG question. So detail about all this disease you will learn later on in the medicine part, but these are the genetical defect which are linked with the biochemical aspect. Okay, so if you understand the metabolic concept, it is very easy to understand the symptom part of any disease, that why that symptom is occurring in this patient. So based on that you can frame your treatment by giving supplements or by giving some prophylactic measures. Okay, so as a long question you have to remember the beta oxidation, can be asked in the 10 marks. In other oxidation you have to remember the disorders. Okay, where we have talked about various acidureas. Okay, Repson's disease. Okay, Jamaican vomiting syndrome, methylmalonic aciduria, etc., etc., and these two, Zellweger syndrome and X-linked adrenoleukodystrophy. Okay, so please remember this particular pathway of beta oxidation. It will be slightly difficult to remember. So whenever you are going to see this particular video, open the book and correlate the things. Definitely it will helpful in understanding the topic. Okay, so that is this much is enough for today. Thank you very much.